clone conundrum, science fiction meets reality. In the realm of scientific advancement, few topics capture the imagination quite like human cloning. It's a concept that has long been a staple of science fiction, from Brave New World to Star Wars. But is human cloning still confined to fiction? Or are we on the brink of turning this concept into reality? The idea of creating an exact genetic copy of a human being is both thrilling and terrifying. It promises incredible scientific breakthroughs, but also raises profound ethical questions. Human cloning isn't just about duplicating people, it's about understanding the essence of life itself. As we embark on this exploration, we'll separate fact from fiction, delve into the science, and ponder the future. Are we ready for a world where human cloning is a reality? Let's find out. Decoding cloning, the basics of biological duplication. At its core, cloning is the process of creating an exact genetic copy of an organism, cell, or DNA fragment. It's like nature's own copy and paste function, but with the building blocks of life. In the natural world, cloning isn't as rare as you might think. Many plants and single-celled organisms reproduce through cloning. Even some animals, like certain lizards, can produce genetically identical offspring. Scientific cloning involves taking the genetic material from one organism and using it to create an identical copy. This can be done with individual cells or entire organisms, resulting in a clone that shares the exact same DNA as the original. While clones share the same genetic makeup, factors like environment and upbringing can still lead to differences. Just like identical twins, clones would be unique individuals with their own personalities and traits. Therapeutic versus reproductive cloning, two sides of the same coin. When we discuss human cloning, it's crucial to distinguish between two main types, therapeutic and reproductive. These two approaches share some similarities in technique, but their purposes and ethical implications are vastly different. Therapeutic cloning aims to create cloned embryos for medical research, not to create a full human being. Instead, it's to harvest stem cells for potential treatments, which could replace damaged tissues or organs and help cure diseases like Parkinson's or diabetes. Reproductive cloning, on the other hand, is about creating a genetically identical copy of an existing person. This is the type of cloning that most people think of when they hear the term, and it's the most controversial. The idea of duplicating a human being raises numerous ethical and moral questions. Both types of cloning use similar techniques, but their end goals and the debates surrounding them are quite different. As we continue to explore this topic, it's important to keep these distinctions in mind. The ethical landscape of cloning is complex and multifaceted. Section 4. SCNT, the blueprint for cloning. Somatic cell nuclear transfer, or SCNT, is the primary method used in cloning. It's a complex process, but understanding it is key to grasping how cloning works. Let's break it down step by step.
First, scientists take a somatic cell, any cell in the body except sperm and egg cells. They remove the nucleus, which contains the cell's DNA. Next, they take an egg cell from a donor and remove its nucleus.
The nucleus from the somatic cell is then inserted into the enucleated egg cell, which now contains the complete genetic material of the organism being cloned. The egg is then stimulated to begin dividing, just as it would if it had been fertilized by sperm.
If the goal is reproductive cloning, this embryo would be implanted into a surrogate mother to develop. For therapeutic cloning, the embryo would be used to harvest stem cells. SCNT is a powerful technique, but it's also incredibly challenging, with low success rates and many failed attempts before a viable embryo is produced.
Section 5. Dolly the sheep, a woolly pioneer. In the world of cloning, one name stands out, Dolly the sheep. Born on July 5, 1996, Dolly was the first mammal cloned from an adult cell. She was a global sensation. Created using SCNT, scientists transferred a fin-Dorset cell nucleus into an enucleated egg. The embryo was implanted into a surrogate Scottish blackface. Dolly's birth proved mammals could be cloned from adult cells. This sparked debates about cloning ethics. Dolly died young, raising health concerns for clones. Her legacy paved the way for advancements in cloning technology. Section 6. the ethics of replication debating human cloning. The prospect of human cloning ignites passionate debates across scientific, ethical, and religious spheres. It's a topic that forces us to grapple with fundamental questions about life, identity, and the limits of scientific progress.
One major issue is the concept of individuality. If we can create genetically identical humans, what does that mean for our sense of self? Critics argue that cloning could lead to a loss of human dignity and uniqueness. Supporters counter that genetic makeup doesn't determine who we are as individuals.
Another concern is the idea of designer babies. If we can clone humans, could we also manipulate their genetic makeup? This raises fears of creating a genetically superior class of humans.
There are also practical ethical concerns. The process of cloning is still highly inefficient and often results in failed attempts or health problems for cloned animals. Is it ethical to apply such a risky process to humans? And what about the psychological impact on a clone, knowing they're a genetic copy of someone else? The ethical landscape of human cloning is complex and multifaceted.
Section 7. Cloning's potential. Medical marvels and moral quandaries. Human cloning presents incredible potential and significant challenges. Imagine growing perfect genetic matches in a lab, or curing genetic diseases by replacing faulty genes.
But the road is fraught with ethical challenges. The use of human embryos is highly controversial.
The debate over human cloning is far from over. It will continue to push us to question what it means to be human.